Back in 2000, very few people were doing pancreatic cancer research, and there was a big push in the community, in the scientific community, for cancer researchers to start studying pancreatic cancer. And it's been a resistance to it back then, and still even today, because it's such a difficult organ to study. I've been doing cancer research for about 25 years now, but I've been doing pancreatic cancer research specifically for about the past 10. Mostly because of the challenge of it. It's a very difficult disease to study, and it's one of the worst cancers there is to a patient. Now, a lot of people took up the cause, including me. I wasn't doing pancreatic cancer research before, and then, like I say, the scientific community said, we need more people, and I said, sure, let's try that. I'll take all the information that I had from all the other cancers I was studying and put it into pancreatic cancer, and after that I got pretty obsessed with it.
I'm studying the role of proteases in pancreatic tumor formation, and using our model for pancreatic cancer. And by knocking out these proteases, we can study what effect that has on the pancreatic tumors forming. What I do is take these mice, and I take the pancreata from these mice and I analyze them using different histological stains, different markers for tumorigenesis, and try to figure out exactly what's going on in the presence of these proteases, and then what happens when we take them away. If the tumors still form, or if they get worse, whatever changes happen, that's how we figure it out. I just got really interested in understanding why and how these diseases exist and how they affect the body and result in death.
Now we're at the point where we understand a lot more about how pancreatic cancer forms, and we know a little bit more about why it progresses and why it becomes more difficult to treat as it becomes further along in the patient. Most of my work is trying to decide, or try to figure out, why the tumor forms in the first place and what are the signaling molecules that tell it to make a tumor. But there's been a lot of progress all the way to the end of life, at least in the animal models, that we are just now starting to apply to humans.
Fibrosis in the pancreas could be potentially bad when it comes to chemotherapeutics because of drug delivery. The fibrosis essentially creates a barrier around the tumor, so that it's possible that drugs can't be efficiently delivered to attack the tumor cells. What we found in our mouse model, when we knocked out our protease of interest and induced cancer, or tried to induce cancer in these models, we found that less tumors formed, and on top of that, even less fibrosis formed compared to mice that develop tumors normally. So that's a positive aspect to what we're studying.
Right now what we've found are a number of molecules that control the ability of the pancreas to respond to injury. And those same molecules that are involved in the injury response of the pancreas are also cohorts in making pancreatic cancer form. So what we're doing is using, again, mouse models of pancreatic cancer, and we're removing these molecules, these genes that make these molecules one at a time, and try to find exactly what they're doing and exactly what their role is in both the chronic pancreatitis, that is inflammation of the pancreas, and in pancreatic cancer. And it seems like virtually everything that we look at that blocks pancreatic cancer also blocks the pancreatitis, and vice versa. It's well known that that a molecule known as the EGF receptor is involved in pancreatic cancer, but most of the time it's been looked at as a molecule that makes the cancer more aggressive. We're looking at what it does before the cancer even forms. And what we're finding is that it has a very major role in allowing pancreatic tumors to form, at least in mice, and we think this will translate into people. And so what we're seeing here is a histological stain that is using an antibody to stain for the EGF receptor in a pancreas. Now this pancreas has acute pancreatitis, that is acute inflammation due to just some minor injury to it. And what we see is that the EGF receptor is upregulated in that situation, which is already known to happen in people. So the mice are mimicking what happens in people. Animal costs run about 100,000 a year. And of course that only gets bigger. I mean, every year the animal costs increase just so that the people taking care of the animals can have a salary. So there's never a case where there's a discount. So it's only getting more and more every time. And of course, as my lab expands more and more, as we get more and more interesting information, we have a choice. We either expand the animal colony, or we don't pursue what we already know is to be important.
We do the preclinical studies. Anything in the mice and anything that's done biochemistry-wise or screening is all preclinical. And then once it gets to the clinical point, you have to screen your patients and get your patients and treat them. So that takes, I think, like 3 to 5 years to even get through maybe phase 1 or phase 2. It's a slow, grueling process. We still try to get it out as quickly as possible so somebody can hopefully start applying it to human research.
I don't just knock genes out of an animal. I don't just remove genes from the animal. I make my choices about which genes I remove from an animal based on whether or not there are drugs that will already target the proteins made from those genes. So it's possible to, take any gene out of an animal that you want, out of a mouse. But if there's no drug to target it or no possibility of targeting that, really it will give you a lot of information. It's very important work, but I try to draw the line at knocking out animal genes that already have drugs that will block them. That is to say, I can much more quickly, in theory, much more quickly translate my genetic studies into a pharmacological study in a human.
When the pancreas becomes tumorous, it also gets what's called desmoplasia, which is an immunoreactive fibrotic response in the pancreas. So the pancreas becomes very hard and it becomes heavier in weight. What we do is this picrocereus red stain to stain just the collagen, the collagen fibers that surround the tumors, the pancreatic epithelium. So when you look at the slide, the collagen will be red and the epithelium will be purple. So you can see just how much fibrosis you have in the pancreas.
I have a long history. My family's been stricken with cancer. My mother, my father, my brother died of cancer. And the reality is that one day I realized that somebody does this work. Somebody actually goes into a laboratory, does the research, and makes the discoveries. And I was already a scientific person even when I was a little kid.
I think that people find that admitting that something's wrong, it makes it more real. But the fact of the matter is cancer is real. The symptoms are real. They're there for a reason, to tell us that something's wrong. And if you ignore those things, then you're not doing yourself any good. And if you ignore the people you love telling you, I have a gut feeling that something's wrong, you're not doing yourself or them any good.
If I have the curiosity to do it and I have the passion to do it at the same time, I'm the obvious person to do it. And that's the nice thing about this job is that I love it. I mean, I come in every day, I find out something new. I'm disappointed most of the time because things don't work. But I'm back the next day because there will be a discovery 1 week at a time, 1 year at a time. That's enough for me. I mean, we just need to make progress, and the curiosity drives you. When you're a scientist, it's all about curiosity. But knowing that you're doing something that will help someone eventually it's a drug almost. You get addicted to it. There is hope, there are treatments, and for the next person that gets it, the next generation that gets it, we think that we'll have more and more progress and more cures, and I think there's more than just hope.
A lot of people in my life have been affected by cancer. Back in 2005, my mom was diagnosed with breast cancer, and that was my senior year at Fordham, and it was right before I applied to graduate schools. I took the GREs, and she told me the night that I took the GREs, and unfortunately since then she's passed. But that's why I chose cancer, because it's personal.